So let's just get started. It's all about enzymes. You guys have probably heard the term before, but probably don't know a whole lot about them, besides that maybe you know that they help speed up reactions. So they're involved in something called our metabolism, which is just all the sum of all the chemical reactions in our bodies, but in each cell. So every chemical reaction that's occurring contributes to this metabolism, and the enzyme is what helps those. So we have a couple just little terminologies things to work through first. Anabolic means to build up, so it's a chemical reaction where things are being brought together, right? So we're adding something to come together. Anabolic, you can remember that as, anabolic steroids makes people really big. Catabolic, it's not the same thing as cannibalism. Cannibalism is eating people. Catabolism is breaking apart. So I guess, same root word. So breaking apart. Other things too, when we talk about substrate, that just means the reactants that are going to be broken down. So these things are the substrate. Enzyme and substrate. So those are our reactants, or the substrate is, sorry. This is called the enzyme substrate complex, and we'll have a little visual here in a second. And then finally, this is going to be the enzyme and the product. Notice the enzyme hasn't changed. It's the same in each situation. The only thing that's changed is our reactants have now changed into our products. So it looks like this. So here is an enzyme, and we'll talk about what the active site is in a minute. The substrate binds to form a enzyme substrate complex, and then it releases our newly formed products. Okay. What I'd highly recommend is just taking 2 minutes, pause it here, and just copy down this diagram, making sure that you can see that they fit for a reason. They're fit based on their shapes. Okay, so pause it now. Enzyme function. So the main thing what an enzyme is going to be doing is involves an active site, where we just saw this picture where we have this specific shapes. That is called the active site, which is where the chemical reactions are going to take place. They're very specific. The substrate fits into this active site in a type of lock and key. Each enzyme will have specific molecules that are going to break down. So for example, something like lactase, which breaks down lactose, like in milk, will not break down sucrose. It's a different enzyme or a different molecule. Okay, so it's very specific. Hopefully I'll just erase this here. So something called induced fit. Even when they come, we have our little shapes like this, they're not going to fit perfectly. What happens is they actually have to squeeze in, and they actually have to push it. Anybody know those little doggy thrower thingy-majiggers? I doubt that's the actual name. You put the tennis ball in there and you kind of got to squeeze it in so it clicks in order to throw it. It's the same sort of thing where the enzyme has to kind of be flexible in order to be able to have that substrate be able to stick in there and bind. Alright, so a couple reactions here. Our first one's called, is called a synthesis reaction. So this is similar to the reaction that you just drew. So if you've already driven it or drawn it down, you don't have to do it again. So here's our enzyme. I'll make them really obvious shapes. Okay, what's going to happen is we have our color change, please. We have our shapes coming. They're separate now. They're going to come along and they're going to join this active site so that it looks like this. Right, so our little shapes are going to be perfect, and they fit nicely in our active site. Okay, and then finally, when it comes back together, it releases our substrate, and now it's a newly formed product. So now these shapes are connected together as one molecule. Okay, so that's a synthesis, and to synthesize means to create. We have this anabolism. So the anabolic reaction. Okay, so if you haven't written that down already or drawn it, you can draw it now. It's really just the reverse. So here's a couple examples, and I'd pause this just to get a couple, maybe just these ones here. Just some examples. As you can see, they all have the ASE ending. We talked about this in class the other day. ASE is just going to indicate that it's an enzyme. So it's nice for the most part, the ASE and the root word will tell you what it's going to be digesting or breaking down. So lipase will break down lipids. Maltase will break down maltose. Lactase will break down lactose. But it's not always like that. Pepsin, which you're going to find in the stomach, is going to start to break down proteins or polypeptides. A few things here. It's just the factors that can affect enzyme activity. The first one is substrate and the amount of enzyme. If you have more substrate and more enzyme, what's going to happen is they're going to be able to form more of those complexes, which means that they're going to be able to cause more reactions to happen, which makes it more efficient. Okay, more reactions can occur. Temperature. There's an optimal temperature that it can tolerate. If it's lower than 35 degrees, what tends to happen, as we know, when we get cold, you start really getting close together and shivering, and you're less flexible. That's why people do warm-ups when they play sports. It's the same thing with enzymes. If this active site becomes getting tighter, then that lock and key fit, that induced fit, won't be possible. The next one is pH. As you guys are going to see, we're going to do a pH lab again, like we did before, but this time with enzymes. And we're going to see if pH will affect the enzyme activity. And I'm going to ruin it for you. It's going to, because what happens is if it's too high or too low, then protein will, which is an enzyme, just make sure we know that, it will denature, which just means that if it's all folded up nicely like a protein, it'll come unfolded and it won't have its function anymore. Okay, next page. So pause it if you need to. Last few, inhibitors. There's actually chemicals that exist that will stop enzymes from happening, and I'm going to show you both right away. One's called a competitive and the other one's called non-competitive. So what a competitive inhibitor does is if this is our active site, it will come along and it will fill that spot. So this, we'll put a big I there, it's an inhibitor. So if our actual enzyme that's supposed to, or our actual substrate that's supposed to combine with it tries to attach, it's not going to be able to fit in there because something's already been taken. So the enzyme won't work, so this won't get broken down into its products. The other one is, it's called a non-competitive inhibitor. So it doesn't fill that active site. What it does is it fills, so it doesn't fill this active site. What it does is it fills a different area, like this part out back. It'll fill that, and what it does is when it fills in that spot, it causes what's called an allosteric change. So as you can see, the active site has changed. And so the problem is, if we have that same substrate trying to bond, normally it can bond to this one because it's circle. Can a circle bond in there? No, so the enzyme substrate won't connect and there will be no products formed again. So draw that little picture. We're going to go over this again in class in pretty good detail because it's a pretty big point. And then the last thing here is cofactors. It's really similar to the inhibitors, but these are good. Okay, so they do the same thing. They fit into allosteric sites, and when they do, instead of it causing it to become denatured and not fit, it actually changes it into something a little bit better. So if you have an enzyme that wants to fit in there, when we have a allosteric change, so our little cofactor fits in there, it causes our shape change. Now our substrate can fit in there. Okay, so we have a couple different types. We have organic, non-organic, organic vitamins, heme like in hemoglobin, and then some metal ions like magnesium, copper, and manganese. All of these help to increase the enzyme activity.